In this video, we will go through the different food groups that get digested in our bodies and the food tests used for food groups identification. Let's start by examining the vital components in the balanced diet of any organism. The first group is the carbohydrates. It provides the fuel which allows the chemical reactions of life occur, for example, the cellular respiration. You should know that all carbohydrates are made up of several units of sugar. The second food group is proteins. Proteins carry out different functions in our bodies, such as growth, repair, and act as enzymes, antibodies, or hormones. Protein molecules are made up of long chains of amino acids. Now the last group is fats. Lipids are fats and oils. They are good resources for energy, and they are constituents of the cell membrane and some hormones, and they play a vital role in our nervous system. Lipids are made up of three molecules of fatty acids attached to a glycerol molecule. Now, what are the food tests that we should be aware of? Let's start with carbohydrates. There's two tests for sugars. The first is the iodine test for starch. So to perform this test, in a test tube, we're going to dissolve the food sample in a distilled water, and then we're going to add iodine and shake. If starch is present, iodine turns into a black-blue color, and if not, the solution remains brown. Now the second test is Benedict's test. This is used for sugars. So to perform this test, we're going to put the food sample in a test tube. Then we're going to add the Benedict's reagent to the solution. It is of a blue color, as you can see here. Then we're going to heat the mixture in a boiling water for 5 minutes. If sugar is present, a brick red precipitate forms, and if not, the solution remains blue. Now for proteins, we use the Biarritz test. To perform this test, first we're going to put the protein solution in a test tube, and then we're going to add an equal volume of Biarritz reagent. It is of a blue color as well. If a protein is present, a purple color appears, and if not, the solution remains blue, as you can see here. Finally, the test for lipids is ethanol test. So first, we're going to put a small mass of the food sample in a test tube. Then we're going to add ethanol and shake. If the solution turns cloudy white, then lipids are present. And if not, it's going to remain transparent. Now, what are the safety measures that you must pay attention to? Number 1, use a clean, dry test tube, because a used or wet test tube might be contaminated and might affect our results. 2, don't use tap water. When preparing the food sample, always dissolve it using distilled water rather than tap water, since it contains mineral ions leading to false results. 3, look for color change using a white background to see more easily and clearly. 4, wear safety glasses and gloves because the reagent used when testing food can be corrosive, such as the Benedict's and Biarritz reagents. 5, check the hazard symbols on the bottles being used before carrying out any practical work. 6, when heating the Benedict's solution in a test tube, there's a risk of splashing, so always point the opening of the tube away from you. 7, ethanol is highly flammable, so place it away from the Bunsen burner. Finally, let's summarize and test our knowledge of food tests by answering these questions. If a blue-black color was formed in a test tube containing starch, then which reagent was used? Copper sulfate, Benedict solution, or iodine solution? Yes, iodine solution. If Benedict solution or copper sulfate were added to starch, we would see a blue color because these solutions do not react with the starch. Next, if Benedict solution was added to sugar, what would we observe in the test tube? Brick red precipitate, blue-black color, or purple color? You are right, brick red precipitate. That's because the blue-black color appears only if iodine was added to starch, and the blue color appears only if biuret solution was added to proteins. Now, if a purple color was formed in a test tube containing a protein sample, then which reagent was added? Ethanol solution, Benedict solution, or biuret solution? Exactly, biuret solution. If ethanol solution or Benedict solution were added to proteins, then the solution will turn cloudy white or blue, respectively, since these solutions do not react with proteins. Now the last question. If we add ethanol to an unknown solution and it turns white and cloudy, then what does this indicate? Fat or oil are present, sugar is present, or starch is present. Yes, fat or oil are present. If sugar or starch were present, the solution will remain unchanged because ethanol doesn't react with either of them.